Cancer. Cancer cells and apoptosis. A cancer cell does not respond to normal cell checkpoints, which would initiate apoptosis in an abnormal cell. You can see there's a damaged cell here. In normal cell division, this would lead to apoptosis. If there's an abnormal cell in cancer cell division, it just continues to divide, producing more cells that can also bypass apoptosis. Tumors. Tumors that cause damage to surrounding tissues are called malignant tumors. They are also said to metastasize, systemically spreading the cancer to other areas of the body, causing cells they contact to become cancerous also. Tumors that are not life-threatening or otherwise damaging are called benign tumors. They are not subject to contact inhibition and grow uncontrolled, but they do not infect surrounding cells. Here's an image here of some oral carcinoma cells. Malignant tumors can invade nearby tissues and infect proximate or touching cells. In some cancers, the cells can be transported to other parts of the body and infect those tissues as well, forming more secondary tumors. Once cancer has progressed, it is hard to localize and remove all cancer cells by surgery alone. In this malignant tumor, which you see here, the cells are cancerous and can spread to other tissues and organs. Benign tumors usually remain encapsulated and in their original location, which you can see in the image here at the left, without spreading to other tissues or nearby cells. They are only a concern if the size of the tumor impedes the function of the tissue or organs they are in. Removal of a benign tumor by surgery usually is successful without reoccurrence. Oncogenes. Proto-oncogenes play an important role in regulating normal cell division. However, it can mutate into an oncogene, which causes cell division to take place uncontrollably and not be stopped by cell death or apoptosis. So here's a normal oncogene, which stimulates cell proliferation when needed. There's a mutation here on this oncogene, which leads to inappropriate cell proliferation, and you can see now the growth of a damaged cell, which continues to grow and is not stopped by apoptosis. Tumor suppressor genes. Tumor suppressor genes have an opposite function to oncogenes. Their purpose is to stop cell proliferation and tumor development. When cancerous cells deactivate these genes, cancer cells increase their rate of division and the tumor grows. Cancer cells can metastasize or spread to other sites in the body by deactivating tumor suppressor genes. The most common sites of cancer metastasis are the brain, lymph nodes, respiratory system, liver, and skeletal system. Typically, when someone dies from cancer, it is not the result of the primary tumor, but instead the metastases, the secondary malignant tumors, are the cause of death. Here are some general types of cancers. Carcinomas are cancers of the epithelial tissue. Sarcomas are connective tissue cancers. Leukemias, lymphomas, and multiple myeloma are cancers of blood-forming tissues. One specific cancer you should know about is melanoma, and you should know about the ABCDE method for recognizing a potential melanoma, the most dangerous skin cancer. A stands for asymmetrical skin lesion. So you can see that there's no symmetry. B stands for the border of the lesion is irregular. C, color. Melanomas usually have multiple colors. D, diameter, are moles greater than a pencil eraser. and E, enlarging, so moles that you see growing over time. Chemotherapy and radiation are the two most prescribed treatments for cancers. Chemotherapy disrupts the cell cycle, typically targeting the mitotic spindle formation. Chemotherapy is systemic, that is, it affects the whole body. Radiation is location-specific, directed at the area affected by the tumor. It disrupts the cell cycle by damaging the DNA in the area, and the cancer cells cannot repair themselves and will stop dividing. Side effects of cancer treatment. The typical side effects from chemotherapy are from the damage also occurring to the normal cells, which are affected by the chemicals. This is seen easily in the fast reproducing cells like hair follicles, causing hair to fall out, and of the digestive tract, causing nausea. Blood cancers are also treated with bone marrow transplants. This involves a surgical procedure where healthy bone marrow is removed, usually from the pelvic bone, and transplanted into the cancer patient. A patient may serve as his or her own donor in some cases. This treatment is used for blood cancers because bone marrow produces stem cells, or unspecialized cells, that can divide through mitosis and differentiate into diverse specialized cell types. These cells can produce new, non-cancerous blood cells. In mammals, there are two types of stem cells, embryonic and adult. Embryonic stem cells are found in the blastocysts of developing embryos. Embryonic stem cells can differentiate into any type of cell. Embryonic stem cells are what we call pluripotent, which means they can differentiate into any cell type present in the organism. Adult stem cells act as a repair system by maintaining the turnover of regenerative organs such as blood, skin, or intestinal tissue. They are multipotent, which means they can differentiate into some, but not all cell types present in the adult organism. In addition to cancer treatment, scientists are developing methods for using stem cells to treat other ailments. A trachea or windpipe was grown from harvested adult stem cells. It was used to replace a woman's damaged windpipe. Because the stem cells were her own, there was no chance for rejection by her immune system. Presently, embryonic stem cells have been used primarily for research. Potential for technologies exist, but currently no product has been produced. Promising research using embryonic stem cells include tissue engineering for organ transplants, in vitro models to test drug response and predict toxicity, creation of neurons for the treatment of Parkinson's disease, alternative treatment for diabetes.